Okay, welcome everybody to First Tuesday. We're gonna get rolling here. So we've got Mark Walker here today and myself. Sorry, we're doing a bait and switch here. So Mark's gonna do all the talking. Dr. Walker is a neurologist at the Cleveland VA and the director of the Ocular Motility Lab there. So he's going to share about some of the work we've been doing together in using virtual environments, virtual reality, video games to address ocular motor dysfunction. So without further ado, Dr. Walker.
Thank you. Thank you. Thanks all for coming today. Even though I'm doing the talking today, obviously this has been a collaborative effort with Michael in particular from the very beginning, which now dates about 8 or 9 years, I think, when we first got started. I'd been sort of wanting to explore the idea of using virtual reality for both the assessment and also treatment of patients who have vestibular and eye movement problems. And fortuitously was introduced to Michael, and we kind of linked together. We've been working together since. It's also, I think, a testament to the value of the collaborative environment that we have at CASE and across our institutions, and that also kind of is reflected in our funding history as well. This whole projects got started with a Garver Award from the APT Center, and then moved on to funding from the VA R&D. So I like to introduce, maybe all of you are not as familiar about thinking about eye movements as I am. So just kind of very basically, why do we have eye movements? What's their purpose? There are patients who have problems with neurological problems or eye problems and can't move their eyes at all. And you still might be able to test their vision and find they have 20/20 vision, but they have a substantial visual disability. And why is that? And the reason for that is that unlike the camera sensor in your phone, the retina, which is our sensor of light and image formation, does not have equal resolution across the entire retina, which you may already know. This is an image of the retina, basically taken with a camera looking through the pupil. This is the view that every ophthalmologist or neurologist will get when they look at the back of your eye. If you've never seen this before, maybe most of you have. This is the optic nerve head where all the axons go from the ganglion cells back into the brain to carry the visual information. Obviously, the blood vessels there, and then at the very center of the retina is this area called the macula, and at the very center of the macula is the fovea, and it's right at the fovea, which subtends about 1 degree of your vision or so, that the photoreceptors, the cells that sense light, are most densely packed. So that's the only part of the retina where you really have enough resolution to see 20/20. So when they talk about retinal resolution, they're really talking about foveal resolution. I think in the periphery of the retina, the resolution is much lower. And you can figure that out if you pick like a word on the screen and look at it and stare at it and don't move your eyes. Ask yourself how much of the rest of the slide can you read, and really you can't read very much because you have to make saccades all over. So the goal of eye movements is to position the fovea at what you want to look at, move it back and forth efficiently and accurately. And if that's just looking at an image like this or a book in the text, then you have rapid eye movements that are called saccades that you just make quickly from one point to another. If something is moving in front of you, then you have slow eye movements called pursuit that will track it. If you move your head, you have to move your eyes back and forth to maintain the direction of your gaze. That's called the vestibulo-ocular reflex. So vestibular eye movement stabilizes the image on the retina when the head moves. And then the other type of eye movement we'll talk about today are verdance eye movements, and that's you have two eyes. It's like having two cameras built into your phone, and you want to make sure they take the same picture, that they're lined up. Otherwise, you'll see double. So the whole purpose of the verdance system is to align the eyes so that both foveas are looking at the same point at the same time. So when I say the word rehabilitation, I suspect that eye movements wouldn't be the first thing that comes to mind for most people. You might think of weakness of your arms or leg, trouble walking after a stroke, maybe rehabilitation from orthopedic injury. But there are a couple of eye movement abnormalities that are commonly treated with rehabilitation exercise. Can I turn the sound off on this because I'm getting a constant echo?
I think it's true.
Oh, that's mine? Oh, yeah. Oh, I thought I had it muted. Sorry, that was me. So the first, we're gonna talk about two of those today. The first of those is the vestibulo-ocular reflex. And that's the first project we started working on. So what's the vestibulo-ocular reflex? I already told you. If I'm looking straight at you and I turn my head, when I turn my head to the right, my eyes go to the left. In my orbits, when I turn to the left, my eyes go to the right. Now I'm always looking straight ahead at you. That's a reflex that goes from the inner ear vestibular sensor organs, the IMU of your body, through the brainstem and then to the extraocular muscles. If the VOR is working normally, then when you turn the head, the eyes always move in the opposite direction. If you're looking at those mountains, you always see them stably. If the VOR doesn't work and you don't generate the appropriate eye movement, so your eyes just kind of move with your head, then your eyes will always lag behind and you'll have to make catch-up movements to get back where you want to look at. And so the consequence is that things will look something like this. And so if you're trying to look at something or drive or read and turn your head, that could be very disabling. That can affect the clarity of your vision, etc. So patients who have infections of the inner ear or inflammation or injury from a head injury, for example, can have damage to the vestibular organs, which are right here inside the temporal bone in the inner ear, that causes them to lose the ability to sense and respond accurately to head motion. And so what we try to do is retrain the brain, basically use exercises to train the brain to reestablish this reflex. And so the common way to do that, sort of classically and still a lot clinically, is basically like any kind of rehab. You kind of start with the problem and you try to kind of work with them where they have the problem. So the problem is stabilizing vision when you're looking at something and turning your head. So the exercises are to do exactly that. So I could, most simply put a big X on the wall or put a word on the wall, try to look at it, turn your head back and forth, and it will move around and be blurry because of the problem we just showed. And you just keep working on that with slow head movements and then fast head movements to try to retrain the reflex to move the eyes appropriately, as is taken here from an example of a handout you might give to a patient and how to do these exercises. The problems with that are you can't really control the therapy very well because you can't control how fast the people turn their head. You can't be sure if they're really looking at what they're looking at. It's difficult to monitor their compliance with the exercises, whether they're actually doing them. You send them home, do all these exercises, come back a week later, they're better or they're not better. Did they really do them? For how long? How well did they do them? You have no idea. And the other thing, which maybe at first is a little difficult to understand, but basically you're kind of forcing them to fix the VOR to normal all at once. Like you give them something on the wall and they have to turn the head. The only way it works is if they entirely normalize the reflex. Like if you have somebody with a weak arm and you take them into rehab and you give them like a 50-pound weight and say, start lifting this, that's a little difficult. You got to start with what they can do and make it a little harder each time. So as we'll show you, virtual reality and computer games, the way we've kind of designed them, have a way to sort of address all these deficiencies. So because we can write a game that a patient will play, that game can record what they're actually doing. So we can end up with a log of their compliance with the exercise program. We can customize the difficulty of the exercises. I'll try to explain that a little bit more in a second, to their particular level of deficit. If they have really bad vestibular reflexes, then we try to make it, so that we're mostly helping them stabilize vision. Then we just make a little bit that they have to add to it. And then we keep making it more and more difficult as they improve is the idea. And we can standardize the exercises that they do, etc. And then we can give feedback to them how well they did, and we can also give feedback to the therapist they see the week later. You know, did they actually do the exercises? Are they making any progress, et cetera, to understand, you know, their response to therapy and kind of what to do next. So with this goal in mind, and this takes us back to the original Garver board, which was about 2017 or so.
Yeah.
We designed, you know, with Michael's supervision and the help of students, a game based on sort of a soccer scenario where the player is the goalie, basically. It's relatively simple as computer games go, obviously, but it implements these particular, you know, strategies that we're talking about. So imagine you're the goalie. This is just a simulation. In the game, that person there would be you. And we started out doing this on a monitor screen, but now we do this in virtual reality. So you're looking ahead at a ball. The ball is going to come towards you, and you have to obviously catch the ball to prevent the goal. And in order to determine which way the ball is going to go as it moves toward you, you have to see a cue, basically, that's given to you only while your head is turning. So let me show this to you, and then we can sort of like parse out what it actually does. So he turns his head the arrow just told him he needed to turn his head to his left. Then he turned his head to the left. And as he turned the head, you might have seen that there was, if you look closely, a little seed flashes only for, you know, less, like maybe 100 milliseconds. And it flashes only while his head is moving. So the key thing is here, he has to be able to see the orientation of that letter while his head is moving. And that only works if he's stabilizing that image of that letter on his retina, on his fovea. So that's a way of making this depend on having a fairly intact VOR. And then his job is to indicate with the joystick which way the C was pointing. And if he gets that correctly, that's sort of the way the ball is going to go. And if he gets that orientation correct, then he has to save the goals prevented. If he misses, then, you know, the ball passes and the goal is scored against him, and the game keeps score. So very simple scenario, but a way to then link the performance of the game to the ability to stabilize your vision when you turn your head. So again, because when he's turning his head, now this is on a monitor, remember, or you can imagine this is kind of what you see in space as you're inside VR. It's perfectly still. It doesn't move. So that's the same really as putting the C on the wall almost, except that you can only see it when the head is turning. But how do we make this easier for him? We move the image with him. So wait till the next one. It should show that. So that was like the full demand. The image is still. But if the whole thing moves with him, then basically we're making, do you see how the whole image moved with him? So I'm just gonna let this play so you can keep watching it as I talk. I guess that makes more sense. Takes a little bit to think about this, but if I turn my head like this and the C just appears, then I just have to stabilize it fully just like I would normally. But if the C moves with me, I don't have to stabilize it at all because it's always in front of me, even if my eyes don't move. So I stabilize it partway, which means I'm doing part of the work and he's doing part of the work. He has to stabilize it, the rest of it. So we might make it 50% of full response that he only has to do rather than the full one. So reduce that weight to 25 pounds or something like that. So we can control that and we can adjust that individually for each patient, and we can advance that along as they progress through therapy is the idea. So we kind of spent the first bit of time just designing this game, getting it all to work, you know, validating the technical aspects of it. And then to test its ability, you know, the likelihood that this is going to be helpful with therapy, we did a study first, and this is what I'm going to show you, in which we tested its ability to modify the reflex in people who have normal function. So, I mean, the whole idea of therapy being effective is that, you know, you have to, that it's inducing brain learning, basically. And in the reflex, if we're able to induce brain learning in someone who has the normal reflex, then that we're kind of at least invoking the the mechanisms in the brain that contribute to learning and gain of VOR adjustment. So let me just introduce this word gain. I mean, you all know what gain is from engineering, but it's the same thing in the VOR. The input is the head movement, the output is the eye movement. So if I turn my head like 20 degrees to the left and my eyes go 20 degrees to the right, to compensate for that, that's sort of a gain of -1, but we usually ignore the minus and just call that a gain of 1. So what we can do, and people who have reduced VOR, they turn your head 20 degrees to the left, they might only go 10 degrees to the right. That means they have like a reduced gain, like it's only half what it should be. That would be a gain of 0.5. And we want to increase that back toward normal. So the question is, can we also increase the VOR gain in somebody who already has a normal response? So you already have a gain of 1. Can I make your gain 1.2 or 1.3 so that your eyes actually move more than they would normally would? And that's what this experiment is all about. So the way we do that is we play the same trick, moving the scene, but we move it in the opposite direction to make it harder. So remember, if we kept everything still while you turn your head, that's your normal response, gain of 1. But what if you turn your head to the left and I move the image to the right? Then you have, if I turn my head to the left 20 degrees, move the image 20 degrees this way, then my eyes have to move 40 degrees to stay on that image. So we don't do it by that magnitude, but that's the idea. So it looks more like this. The head goes to the left, the image goes to the right. And therefore, we are sort of telling the brain that it has to move the eyes by more than it would normally do in order to maintain the stability. And for this sort of experiment, when you're doing VOR learning experiments, you do this for, in this case, we did it for about 30 minutes. It was really 300 head movement trials divided in right and left direction. And what we found was, and this is one simple example, that it was fairly easy then. This is a good example, but it was true, and I'll show you across the group as a whole. It was fairly easy to increase the gain of the VOR. So how do we know we've increased the gain? Well, we test it in the dark. So the way the vestibular reflex works, you don't have to see anything. You can just turn the head and the eyes will move, even if there's nothing to see. That's the baseline vestibular reflex that's not at all involving the visual motion. So when we test it in the dark, before we do any training, again, this is a healthy young person with a good vestibular response, no dizziness or other symptoms, and we just do one of these head movements. Those are called head impulses. And those are short movements, maybe 10, 15 degrees. The scale is not on here, but typically these are like 100 degrees per second. So this head movement is about 150 degrees per second. And so the head is the blue, basically. And then the green and red are, the left and right eye, respectively. And for the ease of comparison, they're inverted. So normally the head would go one way, the eyes would go the other way, but you can't tell if it's the same amount unless you put them in the same direction. So we invert the eyes, let's say. So this would be a head movement maybe to the right, 150 degrees per second. The eyes are tracking very closely, not only in magnitude, but also in timing. And the VOR is like the shortest reflex, so like 6 or 7 milliseconds in most people. And when you turn the head yourself, then even shorter. So this is the normal VOR. Then, this person played the game. They played the game for 30 minutes, did 300 trials, and we measured it again in the dark. And now the eyes move faster by about 28% than the head moving. So this would be counterproductive in real life because this person would have the same kind of problem the vestibular person would have because the eyes are now moving too much, the image would be blurred. But in the dark, we demonstrate the learning. When you do this in a healthy person, then you turn the lights on and they walk around for a few minutes, everything goes back to normal. They unlearn this quickly. But this shows that we have the ability with this to train the VOR and increase its gain so that, that gives us at least some, hope that it has also the opportunity to help people who have reduced function. And as I said, that was one example, but across 18 participants, we found an average of about 15% increase across, between before and after training. And that was true for both active head movements. That means if you turn the head yourself, or passive head movements mean I turn your head. So you have no control of the head movement. You can't do any prediction. And it was true under two different kind of training, paradigms, which I won't go into now, but very robust increased gain, in the VOR. So, then, the next step is we did sort of as a feasibility, then have a few patients who came in to just sort of see how that would work. We only were able to get a few. This was in the middle of COVID, and it's hard to get people to come in on a very often basis, to do this training. Mostly, we're just trying to demonstrate the feasibility. But now we kind of developed this into a home game, and are putting together a trial to use this for home therapy. In the meantime, we've kind of jumped to, as well, in parallel to it, another eye movement problem is commonly treated with rehabilitation. And you might have heard a little something about this if you were here when Luis gave this talk, whenever that was, last year or the year before. And I'll kind of take that a little bit further today. But this is the second problem we talked about, which is convergence. And I told you a little bit about that. Vergence is how you align your eyes to look at things at different depths. Everyone knows if you look at something up close, you have to cross your eyes. If you look at far away, they move apart. The crossing to look up close is called convergence. The separation to look at things far away is called divergence. And therefore, you can really only look at one thing in depth at a time. If I look at you, then everything on the wall back there will be double. I might not pay attention to that. In this case, if you look at the tree, then the mountains in the background are actually double. If you look at the mountains, you see two trees when there's only one there. And that's because, the eyes can only align to one point in depth. And so you're constantly changing that. If you look at your phone and then look at the screen, if you're driving, you look at the instruments and then at the road, your eyes are constantly converging and diverging to make sure that you're looking at that you're focusing both eyes where you want to look, and that's done efficiently and without you really paying that much attention or noticing, any double vision. But it's not uncommon for people to have a condition called convergence insufficiency, where they have a lot of trouble converging the eyes and holding that to look up close. That can, occur in a variety of contexts, but has at least been commonly reported as a consequence of traumatic brain injury, even mild TBI. And since I'm at the VA and we do a lot of clinical work with veterans who've had TBIs as part of their military experience, and since a lot of people have TBIs for other reasons, perhaps in other civilian experiences as well, these are common complaints that people have. And just as a reminder, TBI is an extremely common problem. We still don't understand it very well. But, as many as a couple of million in the US have TBI each year, it's estimated. In the VA, in the military, about 500,000 have had TBI as part of their service in the last 25 years. Most are mild TBI. That's basically defined by how long you have some impairment of your brain function after the injury. And most people recover. I mean, you have a concussion, mild TBI, pretty much you recover. But there's a minority, at least, a significant minority of people who have long-lasting symptoms that, are sometimes sort of lumped together with terms like post-concussion syndrome. And they're kind of hard to put together, understand, not because they aren't real, but, a lot of them are sensory things. I have headaches, I feel a little dizzy, I have troubles, with vision. And so, although we understand some of the aspects of this, there's a lot that's not understood. And so we kind of we're wondering if we could use virtual reality also in our approach with gaming, both to understand and possibly treat this problem as well. So this is just a reminder, I say people recover, but if you ask, enough questions, you can see that a number of people who've had a TBI, at least for in the near period after the TBI, but even for a long period, these last bars are up to 12 months, can have a number of symptoms, that again are somewhat nonspecific: dizziness, feeling irritable, having troubles with sleep. Blurred vision is kind of the one we're focusing on here, and double vision. There have been a few studies that have looked at the effect of TBI on near vision and vision symptoms in particular. This is one that was done about the time we started this work, actually, at Landstuhl in Germany, looking at military personnel who came back to be treated there who had had mild TBI. And they assessed visual symptoms, and just by the veterans or the military service members report, having troubles with near vision was reported, by 60% of those individuals, even in a relatively chronic, phase of their injury, and reading as well. And you consider, certainly we read things that are far away, the exit sign, the signs on the road, this screen, but a lot of our reading is done up close. So that's one of the main symptoms that people have when they have troubles with near vision, especially now that you do a lot of your reading on your phone, held close to your eyes. And the idea of convergence insufficiency in relation to TBI is not a new thing, and they found that as well. They measured the convergence sort of clinically. You go to the optometrist or the ophthalmologist, they may measure your convergence by kind of seeing, how close you can see something before it gets double, measuring you with prisms, and they found about 20% had convergence insufficiency. But this has been debated because it's hard to measure this reliably, in the clinic, and there's lots of different methods. It's not very well standardized. So what we wanted to do is apply quantitative eye movement recording to assess this more objectively, since that's what we do in the ocular motility lab. And we've done that. It's nothing new, but a lot of the sort of laboratory setups are fairly expensive and sophisticated, and you can't just take them into the clinics. But VR, and particularly some of the higher end, a couple of the higher end, virtual reality systems now, the main one we use is called Vario, have built-in eye tracking that makes it much more sort of feasible as a portable way to objectively measure convergence. So that's in contrast to what's commonly done if you have go to a concussion clinic, or you have a sideline evaluation or something like that, where they do assess a lot of these functions just by questioning nerves, for example. In this case, the Convergence Insufficiency Symptom Survey, our CISS, just asks questions, do you feel tired when you're reading? Do you get a headache when you do close work, etc.? And then just adds the points all up based on how severe it is on the scale. And that gives a measure of your near vision symptoms. And that's sort of then extrapolated to say these people have convergence insufficiency, but no one's really measuring convergence. So one of our questions also in doing this work was kind of how well are these sort of subjective surveys actually working as surrogates for the objective testing. So we got this started. The main way that you do this that you measure convergence in terms of your eye movement function is something called the near point of convergence. I sort of alluded to that before. If you follow, anybody can do this. If you just follow in, eventually you reach a point where you can no longer converge. For me, it's about right there, and I see two thumbs. In the clinic, you might have a little target that you do, but it's still very subjective. You can watch people's eyes, but you're sort of relying on them to tell you when you see double. But if you do that, you'd see something like this, where the eyes are converging as the target is coming in, and then eventually it's going to break, and you'll see the eyes diverge. And in our VR, or we do it this way with a little sort of green object that's coming closer in, and when the convergence breaks, then you see double, basically. So you're asking the patient, when do you see double? You can kind of look at the eyes, but what we do is then record the eyes, and we can mark the exact time that the convergence breaks and the eyes diverge. So here's an example of this. Let me talk you through this. This is near point of convergence measured in VR, exactly the way I showed you. So this dashed line is the target, just the convergence angle of the target. So if the target is far away, this would be 0, basically. The target is far away, then you don't need much convergence. As the target comes closer, you need more and more convergence. And this is moving at a constant convergence speed, basically. And then when that happens, you have two eyes. The red eye here, or the red is the right eye, and the blue is the left eye. The eyes move in opposite directions. In this case, the right eye goes to the left eye goes to the right. That's how you have convergence. If you subtract them, then you get the green line, and you see it sits right on the target here. So until they get to about here, which in this case, I don't have the scale here, but it's the same recording here. So it's about 40 degrees. Once you get to about 40 degrees, which is probably right about there. So then this person can't converge anymore to that target. Then the eyes come together, the eyes diverge, the convergence angle gets much less, and you see all these back and forth movements, which are the odds you make as you're trying to decide, do I look at that one or that one when you see two targets. So we do that 6 times in a row to get a better sample and average those. In this case, the near point of convergence would be the maximum convergence, so about 40 degrees in each of these, and then average those. In our initial study, then we enrolled 3 groups of people, basically. Veterans who had TBI, who had problems with their near vision that were defined by that questionnaire, so symptomatic, subjective near vision issues. That's group 1, so their CISS score, that survey I showed you, was greater than 21. And then we tried to enroll individuals with TBI who had few vision symptoms, but actually found that they were hard to find. And that kind of goes along, I guess, with the fact that, the prevalence of the symptoms that other studies have found in this group. Also possibly because of the non-specificity of the symptom. And then we compared them to a group of individuals who had no TBI and no vision symptoms. And I guess not surprisingly, we actually found that the survey didn't predict their actual convergence very well. So we measured the NPC, Center Virtual Reality NPC, again in degrees. So a high number is a high angle of convergence, so that's good. A low number means they can only converge to a small angle, that they can only see single at a further distance. The blue ones are basically the controls who never had TBI. These red ones are the ones with TBI who have near vision symptoms, and the yellow ones are the ones who haven't. So, at least in this relatively small sample, it sort of, I think, emphasizes the importance, at least in moving forward, of making sure that whenever possible, that the diagnosis is made by an objective measurement and not just by subjective symptoms, which makes some sense. But it also showed that the few people that we had who said they didn't have any symptoms based on the questionnaire, 3 of them still had relatively bad convergence by the NPC. So it's not only that it doesn't accurately predict convergence in people who have symptoms. There might be people with symptoms who actually converge pretty well, but the opposite was also true. So it was really, the difference was the TBI versus no TBI more than anything else, and that's shown here. This is just a box plot of the NPCs themselves based on whether the group that had no TBI and the combination of the groups who had the TBI. And you can see that there's a lot of spread. Like there are quite a few people with TBI who have very good NPC, but a much larger number who have poor NPC than those who don't have TBI. And if you put everything together, 9 of the 16 TBI individuals had a low NPC, which you defined here in it says 30 degrees here, but we're actually using 33 degrees, but it's really the same for this plot. So even though there's a wide range, there are quite a few people with TBI who seem to have poor convergence, even though doesn't correlate with their own report of symptoms. And so in our next study, we're going to have a much larger group of people with and without TBI to try to sort this out a little bit more precisely. But, NPC is just how far can you move something close before you see double. Life is much more than watching things come close to you and then, not being able to see them anymore. So near vision, as we said, and convergence are very important for things like reading and also things like depth perception. So, if you have 2 things in front of you, how do you know which one is further away? You can imagine it's easy to tell if something is to your left or to your right, because if it's to your right, it falls on this part of the retina. If it's to your left, it falls on this part of the retina. You can see that. But if 2 things are in front of you, there's no way to tell with one eye which one is closer and which one is further away, because they kind of fall right together. And for that, you need 2 eyes, and you need to be able to compare how the images line up for each object on the two eyes, which remember would be different depending on your vergence angle. And the ability to do that is called stereo vision, and to tell, okay, because of, when I see double or not, based on where I'm looking, this thumb is further away than this one. In the clinic, we sometimes, and we test the, how good your stereo vision is with a measure called stereo acuity. It's like, well, if I move these thumbs really close together, how close can I get them and still tell that one is further away from the other, basically? And so in the clinic, we use these little books, and you have like stereo glasses, polarized stereo glasses. They're kind of like watching a 3D movie, and these objects, you have to kind of detect which one is out and which one is relative to the others, basically, and they get closer and closer together. But VR is kind of an ideal way to do this, because much better than a book, which you have to, we have the ability to precisely, and the whole point of VR, the way VR works, is that you simulate distance by the relative positions of the images that are projected to the two eyes. And then, and that's not only controlling the vergence angle, but giving you the perception that something's closer or further away, even though those screens never move. So we can do that to measure stereo acuity in this way. So we use what's called a random dot pattern. So you generate sort of just a grid here, basically, of dots. Pixels are black or white, kind of randomly. And then it's the same. This is the image for the left eye. This is the image for the right eye here on the right. And then we make an arrow that's also separate, but that's also made of random dots. And it doesn't appear as red or blue. It's a random dot pattern, but I'm just showing you the fact that we can put that arrow at different places on the two images. And therefore, the distance that image appears is different from the background, or alternatively, you have to admit your vergence angle is different to line up the two arrows than it is to line up the two backgrounds. And then you can, by that, determine whether the arrow and the background are separate. And then what we ask the patient or the participant to do is to tell us with a joystick which direction the arrow is pointing after it flashes like that. So again, you can't simulate 3D and 2D, but you can imagine it looks something like this if you're looking in VR. So you can see the arrow offset, from the background, but only when both eyes are looking at it and when your vergence is looking well. And so what we do is then we present these sets of images with arrows in background at a range of different virtual distances, really far away, so it should be easy to tell, and really close up. And that's called the disparity. Stereo disparity is kind of the angle difference between the two sets of images. And so really low means they're really close together. Big numbers is really far apart. And then they just have to guess, which way is the arrow pointing? If they get it right, they get a 1. If they get it wrong, they get a 0. And we fit those data with a sigmoid function. And then we can sort of tell where the acuity level is based on kind of where this fit is. And then we can manipulate it in another way, which we can ask them to do that as if the images are really far away. So starting with a vergence angle of 0, if that makes sense. Or we can bring the whole thing forward as if the background is close, and then can they distinguish small distances up close versus small distances far away. So that's what far and near means here. And you can see that this particular individual had much more trouble, or at least, considerable more trouble, and made more errors when we brought everything closer. So it was harder with the eyes converged than with the eyes diverged. And then we asked whether this depended on the TBI or not, in a similar way to the MPC. And this is kind of like doing one of those fits for every participant, and then, putting them together here with means and confidence intervals. And so people who don't have TBI, these are kind of their fits. For far and blue, red and near, they overlap fairly well. And the TBI individuals, you can see they did worse in kind of both conditions, but in particularly when looking up close, when the images were close. So they had much worse stereo acuity. So it's not just that they can't, keep something from coming double while it's coming close, but they lose stereo acuity function, particularly at near, but maybe also a bit at far as well. And then the last thing I'm going to show you here for the assessment is reading. So like I said, if you read up close, you have to really converge well, and to keep those images aligned. And so we did that with these letter grids and, asked them to read just, grids of letters. And we did that again, far away, so that they had no vergence angle. Then we moved it to about 20 centimeters in virtual space. And so this is one participant. I'll just show you quickly, how they did, and you should be able to hear them read here if it works. I guess we went through.
KCRCKVKOZS, ZZNNHVZZKR, KSHDKOSVNH, ZRHDDNRKRH.
Reading accurate. And then we just moved the image together, and then, there was no change in size, anything else that would have been a view that the images were, that it was closer. And then we did the same thing, and had them read that. So let me try to play that one.
NHNKHODOKZ,
Second one.
SVRNRSVRKO, SSVS HSOK.
So you have no idea what he's reading. So we can then, because we're recording the eye movements through that, we can plot out the scan paths as well. So like I said, when you read, you actually have to make saccades across the page so you can see each letter. So that's what you see here. So you start at the top, you make saccades across to here, then you come back, you make saccades across this way, you come back until you finish the page. And here it's just very disorganized, kind of all over the page, jumping around. And that sort of correlates with sort of how he's reading. So again, a lot of disability because he can't keep the images aligned in a way to read up close. So we can, get a little deeper into near vision function by looking at things like stereoacuity and reading, and also mapping out the eye movements during that. And we can theoretically, although we didn't do that in this case, find the distance where they could read, the closest distance where they could read well, and kind of use that also as a way to kind of getting trained. So I know I'm kind of running late here, but let me just say a little bit about how we then use this for therapy. So similar to the vestibular therapy game that we developed, from the beginning, we also wanted to develop a game to help train the convergence insufficiency. And this is usually called, if you go out and get this therapy from an optometrist or an occupational therapist, it's usually called vision therapy. And basically it's exercises they're trying to train convergence. And sort of classically how this is done is, again, relatively primitive. I mean, we call them pencil push-ups, but you just take something, and just kind of look at it, try to bring it close, comes double, you try it again, imagine doing that 20 minutes a day or something like that. And here are the instructions from a handout that came from the DoD actually for this problem. So you're practicing the convergence. You can do it if you do it in the clinic with optometrists, they can do it a little bit more precisely and monitor you and help you along. But again, if you do it at home and you come back, nobody has any idea whether you actually did this at all and whether you did it in a way that's likely to be effective. So we wanted similarly to develop a game that would require convergence to play, that would be adjustable to each person's deficit, that they could do at home, that would be a little bit more engaging than looking at a pencil coming toward you for 20 minutes straight. And that we could keep track of their performance and kind of log that. So we developed a game that, this is kind of a version of it. Basically, it doesn't really matter what the game scenario is, as long as convergence is required. This is a fruit falling game where you have to catch the fruit, basically. So the fruit fall down and you have a basket that you move back and forth. And so these fruit not only vary in their left and right position, but in their distance from you in virtual space. So you have to judge where they are in order to put the basket in the right place to actually catch it. And then you're scored based on, the catching the fruit and et cetera. And we tried, I guess, 3 people with this as a feasibility. And now we've kind of got this in a format on the MetaQuest that can be, or whatever device we choose, but likely that they can play at home. And we have a trial sort of that's supposed to start in October funded by VA merit review that will allow us to test this for home therapy. But here's an example of our, so of one of our participants who actually did demonstrate some benefit seemingly from this. So he came in 8 sessions once a week, so 8 weeks total. And played the game, you know, as we adjusted its sort of difficulty as time game went on. We designed this mainly as a feasibility trial, not really as a treatment trial, so kind of adjusted it as we went along. So it wasn't done with the care that would be part of a clinical trial. But nonetheless, you can see if we just take his playing and just like he plays this game for 20 minutes and then these are just all the vergence, you know, just a histogram of his vergence angles during playing. Remember, low angles are far away, high angles are near, and normal convergence is kind of up here. So he's playing this with kind of lower than normal convergence, and we kept making it more difficult for him. And you can see that the vergence angle range increased, and he had more convergence in session 8, which partly could mean that we're just making it harder for him and forcing him to do that, but also at least that he was able to do that. So then we measured his NPC over time as he played the game each week, and within each session, you know, we measured it at the beginning and the end of the session, and also in the middle. And so here are the results for the 8 weeks of training. So he started out with NPCs in the 20 to 30 range. Remember, we kind of, our lower limit of normal is right now is about 33, and many people are up around 40 to 50. But if you look at over time, he did his average convergence increased, and you know, if you take his beginning one at about high 28 or something, and he went up to almost about 40, that's a considerable improvement. Again, at least as a preliminary effort. And remember, he only played once a week. He didn't do this at home. He didn't do this, you know, on a daily basis. And more than that, he told us, you know, that he was much more comfortable with near vision after doing this as well, holding his phone and everything. So, you know, an N of 1 in an uncontrolled trial doesn't prove anything, but at least this kind of demonstrates the feasibility of this approach. So now our goal is to test this a little bit more rigorously in a crossover trial at home with a larger group. So anyway, I hope this gave a little introduction to at least what we're working on. The value of physiological measurements of functional impairment, whether that's the vestibulo-ocular reflex or the ability of convergence, are better than just subjective symptom assessment. It may give us a better understanding of mechanisms and how these ocular motility deficits impact, you know, normal behavior. Virtual reality has a lot of advantages for diagnosis and treatment of ocular motor dysfunction, and you know, that we talked about before. We can customize it. It's more interactive and engaging. We can use quantitative eye tracking to monitor performance, and it has the portability. Hopefully, that will allow us to deploy it a little bit more widely. And thank you very much for attention. I'll take any questions if you have on Michael. Michael's responsible for most of the technical aspects of this, so we can both take questions.
Yeah, any questions?
Just watching Zoom.
Oh, yeah.
Any questions, but,
Yeah, Lindsay.
Yeah, can you comment on whether it's possible to use similar techniques to help with bigger problems like reading, as you age, you need glasses or double vision, from one eye, the other, the other one. Can you use the same virtual?
Oh. So I mean, there are two sides to that. One is the assessment side, obviously, and the other is the treatment side. And you know, certainly we haven't tried. I mean, certainly I'm very interested in that question. I mean, the first step we're taking in that direction is that we're in our next study, the recruitment goals involve recruiting individuals who have convergence problems that are unrelated to TBI, so we've never had TBI. It's a fairly common problem in general, and also over a wider range of ages as well. In this study, I skipped over the slides, I guess, but we only enrolled individuals up to 45. So we might, we might, incorporate, or we might be able to sort of get some information on sort of aging related issues too, although this is still specifically targeting, convergence. Some people have double vision for other reasons. We don't do a lot, I mean, there's not a lot of vision therapy for other reasons for double vision, like people will have injuries to a nerve. One of the nerves that moves the eyes, it's a fairly common problem. A lot of times those do improve, but they don't, they're treated with prisms that sort of optically align the images in glasses, or they're treated with strabismus surgery that kind of attempts to realign the eyes. Maybe some exercises, but I think the advantage of this, potentially, both in terms of the assessment, in terms of you can find where people are in terms of sort of match their eyes and align everything, and then try to move it from there. So it'd be sort of interesting, I think, to extend this to some of those other eye movement problems, other causes of double vision, and see if those might also be, amenable to this kind of rehabilitation, but.
I think strabismus, there's interventions for pediatric strabismus using VR, also.
Yeah, I mean, certainly there is some work going on in that too, that's right, for pediatrics.
So maybe the key point is in the VR, everything's at a fixed distance. And so we're not actually physically changing, the distance. And you didn't mention this, but accommodation is something we're not able to train because it's at a fixed distance. Yeah.
But we do have the advantage, you can manipulate the images however you want.
Yeah.
So it has much greater flexibility potentially for some of these other problems.
Right. Any other questions or thoughts? Yeah, Fred.
When you do vestibular therapy, do they have a normal vestibular system and it's not communicating with the visual system, or is it communicating random stuff with the visual system? Or do you know what the problem is there?
When you say what the vestibular system is communicating to the visual system, what do you
I
mean?
mean, when I tilt my head, my eye accommodates,
Yeah,
but
so the bit.
theirs doesn't apparently. So there's some sort of interaction there. My eyeball knows that my head's tipping.
So, I mean, when you turn your head, that's sensed by the labyrinth, by the, essentially the gyroscopes, the semicircular canals in the inner ear.
Yeah.
That information goes to the brainstem, vestibular nuclei, and that goes directly then to the neurons of the nerves that can move the eye muscles to move the eyes.
But that's not working.
That's the vestibulocular reflex. So normally what I'm talking about is when the sensor is not working. So, you have impairment of the semicircular canal function. So you're not getting the right motion signal in. And you have,
So
usually
it's-
that's decreased, you have to turn up the gain of the reflex.
So the vestibular system isn't working.
Right. But it's the visual system then that gives that error signal, that tells you that
I know.
the vestibular system isn't working, because that's the visual feedback you get when you turn your head.
Path is through the brain.
Yeah,
That path is intact.
I mean, there are cases, if you have, for example, the cerebellum has a lot to do with this learning and controlling the gain of the VOR. So there are people with brain problems or cerebellar problems, cerebellar degeneration, that can also affect your VOR, because that calibration gets out. And it's a little harder, at least we haven't tried that with our setup, but that generally is harder to rehabilitate because the learning mechanism is impaired as well.
Yeah, Paul.
So in one of your first experiments with a healthy volunteer or healthy subject
Yeah,
you.
yeah, basically.
How long did that last? And do you think that if you were exposed to that in a VR game or something, that might have longer lasting negative effects?
We didn't, I mean, in these experiments, we didn't measure. Most people don't notice much. I mean, I'm sort of looking for this, like, for me, I noticed like a little bit, maybe a minute or two at most, it lasted afterwards. Remember, what we're doing is, we're taking a very stable, hard-learned reflex and just modifying it in a short term. There's no consolidation of that. There's no, long-term learning there here. So we probably haven't engaged all the mechanisms that would make that last longer. I mean, in the very beginnings of the research for VOR learning, we're done with magnifying glasses that you would wear all the time, even reversing prisms in one case. Wear those for 2 weeks, and it changed your VOR. That was a much longer time because it was continuously for 2 weeks or more. Then it took a lot longer, days or weeks for that to recover back to normal. But if you just move away from what hard-learned normal for 30 minutes, after a 30-minute training, it goes right back.
I just wondered whether it had implications to sort of, no video games and other things are tested for this, whether AR, VR, you have kids, doing, if there was misaligned or poorly programmed, that it would, by happenstance mimic this, whether that you could have long-term consequences.
You could talk about a little bit more.
Well, I mean, I remember when I first asked you about this, when we were working on this, I think you gave a good example of that. I remember when I first got glasses, so wear it early in the morning. And then, cause I would get this blurry vision actually when I'm wearing glasses. And then eventually, after a week or two, it goes away. And I remember asking about this and you said, well, yeah, you have these modes that the brain sort of remembers and can, so when you put on your glasses, or you take off your glasses, it's not a maladaptation, it's an adaptation. And so I guess in a sense, we're taking advantage of that ability, the neuroplasticity, to say, okay, well, sure, to Fred's question, that the hair cells aren't tuned the same way anymore, but maybe we can develop a new normal, a mode that, they weren't reaching because they weren't training. I mean, some of these folks, like the guy, the 8-session guy, where he got the concussion somewhere in Middle East and then got sent back to the truck with a bunch of aspirin and never treated it. And then, I don't know, like 10 years later, shows up in our lab and didn't know we could get better. Right, so it's tapping into maybe like stroke rehab or other sorts of rehab, some neuroplasticity potential that we always use, we just don't think about it. You're saying, you look through the zoom lens of a camera, and things are moving faster, and they're not walking around wobbly because they've kind of switched modes when they put that camera up to their eye,
But to get back to your question, I mean, for there to be a problem for a normal video game playing, there'd have to be a lot of head movement for that game. Because this is all about moving the head. And then if you had like head movement and laggy, I mean, for the VOR, if you don't move your head, then it doesn't have anything to do with.
And it is, I mean, some people we've run into.
But when you do get
nauseous.
But we, where you could get problems are sometimes if it's laggy.
Yeah,
You have a lot of visual vestibular mismatch if it's laggy.
that's the medical sickness.
So, because that's not necessarily a gain problem, but it's a timing problem. The image doesn't keep up with you. Sorry, I forgot what I was gonna say, but, things are, if the environment is moving, if you've ever been to like, I don't know, my family went once to Universal or something, it was this Harry Potter ride, this virtual reality or whatever. And they really make it feel like you're moving, but you're not. So that's another case of visual vestibular mismatch. So if the game has a lot of simulated movement, but you're not moving with it, that also is a problem.
That's what
It's not really,
on that. But yeah,
I'm interested, sorry, did someone else have?
go ahead.
As you upsize these clinical trials, what sorts of covariates would you control for? I was just thinking about age being one, with younger people having that increased neuroplasticity, or just having more familiarity with playing video games where they might already be able to be making those adaptations. Are there other variables that you would control for in those analyses, or?
yeah, I mean, obviously the number of things you can control for depends on the size of your study, really, so. But,
We did it because we
I
want,
mean, keep in mind that like maybe someone there is rehab, like despite the fact that these vision therapy exercises are done, like there's very little data actually to begin with. So even now with our new study, I mean, we're kind of still just at the very preliminary sort of feasibility, preliminary efficacy thing. But yes, we would definitely control for age. I mean, we're looking at TBI, but I think your point is well taken, that there's a lot of other factors, that would potentially influence just their efficiency of learning.
Yeah, we do have
We
to
have
ask
to
those
look at, so,
questions.
we do ask those questions as much as we can, but I don't think we'll have a big enough study yet
No.
to control for all those things.
Do you have, have you used VR, things like that?
Yeah.
I think how often you play video games, things like that. We ask anyone.
Yeah,
Yeah, so this is somewhat going back to 2 questions ago, but I was wondering if for the population of TBI, if using the virtual reality increased any of their other symptoms, and if so, how does that compare to other measurement or rehabilitation methods for TBI?
Again, so our experience so far with that game with TBI is very small, right? The next study will give us a little bit larger sample size, but,
we asked them.
we
after
did it like a
every-
scale of,
Yeah.
nausea mainly when they were playing, and we asked them about other symptoms. We didn't do that really super systematically. But, they all tolerated it well, like they
Every
all
100
came back, they all had the option not to come back, and they came
Right.
back for at least 6 sessions. All of them, and participated again, and completed, completed the sessions as well, so.
I think on a scale like 1 to 10.
Yeah.
They would be up to a 3
Two
sometimes.
to three, something like that, so, but that was like during the playing.
Yeah.
You're asking like if it could just like alter other baseline symptoms or something, is that what you're asking, or
I guess just in
make their headaches worse or something?
Exacerbating, yeah.
Yeah, I mean, certainly
so-
that's possible.
Light sensitivity is one
yeah.
thing we tried to
Mhm.
tune in this, so we gotta like lower things down
Yeah.
as much as possible. Nausea, vertigo. Thankfully, nobody had more than 3 out of 10 across like the 300 trial. So we'd break every 100 trial we passed. Nobody.
I also had a second question, sorry if you remember this, I came a little late, but for the TBI population you worked with, what was the general range of time since the injury?
So very long, actually.
Yeah.
These are all veterans that we see. I mean, I can't give the
Years.
exact number where I have it, but
Yeah.
we see these are the people I see like in the TBI clinic, and some of those, their injuries were like 20 years ago. So again, we're hoping to, I mean, that's the other goal as this moves forward, is to, pull in more people who have recent injuries.
Yeah.
I don't think we can say a lot very, definitively about the TBI and convergence insufficiency, relationship just based on this study. For some of those reasons, I mean, the TBIs were quite remote.
I mean, we did
But-
take it over to Metro and, showed it to people who had just had, motor vehicle accident in the last
Yeah,
couple weeks or something, and that's, that was more of a tolerance thing too, right, to make sure.
yeah.
Yeah, they don't, they can handle that, but yeah, the veterans were at least
Yeah, and
2, 3.
we didn't
years
test
out.
any of those in terms of, their treatment or anything.
No, we couldn't yet, but at least we could tell, that they could, in a sitting position, tolerate. it. Yeah, but the lighting is a big issue.
Like,
Yeah. And since most of this is not, well, the convergence insufficiency one, they're not moving their head, they're just sitting still, and we're moving objects, for them. It's a little bit less sensitive, but yeah, the vestibulocular one, we didn't have any issues with either, but that was back when it was on a TV screen.
yeah,
Yeah, in a dark room before the VR headsets got more affordable. Yeah,
Did you keep track of the best uncorrected visual acuity range of all your subjects?
yeah, we have the data, they did it with with correction.
Right. But,
so you did not notice any difference between people who are, say, high myopes versus people just guard presbyopia?
Okay, well. Presbyopia, we don't have much of on because most of these were younger, but, I don't think we don't have enough sample to sort of. Yeah, I don't just think we have any high miles, maybe one or two or something like that, but not many.
What is the virtual focal plane distance, I think with these MetaQuest?
I think most of this was on the Vario, actually, that's like. That's hard for us to figure out, actually, we've been trying to figure that out, because there's a lens in there.
I mean, it's more or less focused at distance, but we were talking about this the other day. like,
Yeah.
What is actually the focal distance?
Because,
Not sure.
The Apple Vision Pro was about 4/3 meter, about 133. So they ended up putting in a 3/4 correction for that.
Yeah, that was the one where you have to get the
Yeah,
custom
you
lenses,
get-
right.
Yeah.
But I mean, this one, they more or less focus it to far away, but I don't know if that's really officially infinity or,
I mean,
2 meters
we
or
went as
10
far
meters.
as like the room was big, right? We took like a controller over here and,
Yeah,
kind
but
of
it's a little,
physically measured.
Because around a bit under 5 feet is about where virgins really don't really have much convergence.
but see, convergence, the thing about VR is convergence and focus are separated. Because convergence is just based on how you move the images to create the disparity.
Does the lack of accommodation
But
mess with anything?
no, because like I said, that's a question, I guess, I mean, there's pluses and minuses, right? We can test vergence independent of accommodation, but we're not testing vergence facilitated by accommodation, which I think the question that you're asking.
Yeah.
So, trying to make some comparisons between, real life and virtual reality as well, and. But I think haven't fully answered that question, but, and of course, for older folks, then the accommodation is, there's still blur, but the accommodation is less. I was talking to Bottom about all this the other day too, so.
All right, well, thank you so
Yeah,
much.
Thank you.